So next, I think our third speaker, Clarissa, has come back. So Clarissa?
Hi,
Yeah.
thank you for waiting. I'm sorry for the technical difficulties. My Zoom, they just decided to fail. But
Yeah.
I hope it's okay now. I'll try to share my screen again. Hopefully it's showing up now.
Yes, I think it's working. You can start it right away.
Is this correct?
Yeah.
Is this
Yes.
good?
Yes.
Okay.
Yeah.
So great. So thank you for the organizers of these conferences, for inviting me to give a talk. And good afternoon or evening, everyone, whenever you are in the world. My name is Asha, and I'm from Indonesia. I'm currently living in the UK. I'm a PhD student at the University of Cambridge, studying biological anthropology. A little bit of introduction. I've actually been a member of Dr. Safarina Malik's lab in Indonesia since 2011, and I've been assisting her with her email project, which we developed together with Andy Holmes at the University of Sydney. So I would like to start this talk by building upon what they presented yesterday about diversity and biogeography of the microbiome. So biogeography is essentially a study of variations in life across time and space. In this case of the gut microbiome, studies have shown that it is a diverse entity. And as we have seen through all of the talks that we had from yesterday and today, there are variations in the structure of the human gut microbiome. And what I'm highlighting here is the study by Costia et al. Here, which shows the enterotypes of the human gut microbiome. And the structural diversity has been associated with geography as well. And the second study I highlighted here on the top right is by Fanjai et al., published in 2018, which is about a multi-generational drift of the gut microbiome, in which the microbiome changes rapidly in immigrants that moved to the US, and that change become more pronounced in later generations. And the third study on the bottom right, I highlighted here, is by Smith et al. In Hadza hunter-gatherers, which shows that there are seasonal changes in the gut microbiome. And basically, all these studies demonstrate that the complexity of the gut microbiome and its variations, which can be observed structurally and be inherited through time and influenced by competition for resources in changing environment. So all these three aspects, variation, inheritance, and competition, place the microbiome in the framework of Darwin's evolution theory. And thus we can use this framework to ask some fundamental questions. In general, there are 4 questions about evolutions that we as a scientific community, I think, need to answer right now about the gut microbiome and its evolution. And that is, who is involved in the process? Why did it happen? How did it happen? And is it a predictable process? And in other words, to understand the biogeography in the microbiome, we need to know 4 things: the subject, the cause, the mechanism, and the trajectory. So for my PhD study, I'm interested in looking at the population background in Indonesia and finding how it can influence the evolution of the microbiome. And by what I mean by population background here are 3 things. Essentially, they're an ancestry, culture, and ecology. And these factors are interrelated and can influence it. And the influence of each on the microbiome can vary for local populations or individual or locations in different geographical area. And I am particularly interested in the differences between non-industrial and industrial societies. For instance, the type of food that can be accessed from these societies are very different. For example, people living in industrial societies are more likely to live a sedentary lifestyle and sedentary and individualistic, while people in societies that practices hunter-gatherings or subsistence farming will likely have a higher level of physical activities and then are more community-oriented, needing to share resources with others in the community. So the data that I'm going to present today is from a preliminary study of communities in several locations in Indonesia. And the slide shows all the sites that we have visited since 2015. And there are 2 sites in Bali and 5 sites in North Kalimantan. And then there's 1 site in Papua. And Bali is a well-known tourist destination, known for declining population health due to a shift in their lifestyle, while the Kalimantan locations includes settlements of the Punan ethnic group, each with unique characteristics. And the Daikot village in Papua is an interesting group to have here because of their distinct ancestral background. So this is a table that highlights the important characteristics of this community. I colored the last column here to make note of the type of what these communities that we consider. So we have Denpasar, which is a city, and then Singaraja is also is in Bali. Padawa, which is in Bali, and the closest city. We consider this a peri-urban environment. And then we have the Basap community in Teluk Sumbang, which is also considered a peri-urban. And then we have 4 populations of Punan in Kalimantan. They are Respentubu, Riantubu, Long Sule, and the Punan's Batu Saja rock shelters. Of these, only the Respentubu is what we consider a peri-urban, and the others are remote. Lastly, we have the Asmat population in Daikot village in Papua, and it is also a remote location. In addition, we collected data on hunting frequency from these communities, and we found that there are 3 communities with high hunting frequency, and these are Rian Tubu, Long Sule, and Batu Saja communities. So the pictures here on the bottom left is Teluk Sumbang, which is where the Basap people live, and on the top right is a picture of a hunter-gatherer Punan Batu ethnic group and the Punan Batu Saja rock shelters. And here is a chronicle of our data and sample collection efforts since 2015. As you may think, building this collection required tremendous effort and requires multiple trips and many hands from a well-trained and physically fit people here at the Marine Institute. And we found that the key to success was collaborating with the right partners and properly engaging with the local communities. Notably, we visited some location multiple times, and these are the Teluk Sumbang and Punan Batu Saja community. This year, we're also planning to return to Respentubu and Long Sule to collect more samples and conduct a more thorough anthropological survey. And this will allow us to observe for changes of the microbiome over time in these communities. Along with the way, we did like 16S analysis and metagenomic sequencing as well. The metagenomic sequencing particularly is only done, it has been only done for 140 samples, but we're expecting about 10 million reads per sample for this sequencing in sense. So I published a paper last year about the results of the 16S analysis in the Balinese in Denpasar community only. And the take-home message is here is that there are 2 types of community in the Denpasar data set. And these are, one of the communities dominated by the Brevotella, as you can see here, shown in light blue. and the other half of the community is dominated by Bacteroides, which is shown here in dark blue. And when I analyzed the Denpasar data in comparison with data from other populations, such as the Hadza hunter-gatherers and Americans, I found a unique pattern. We can see here that the distribution of the Prevotella and Bacteroides is equivalent in the samples from Denpasar. Typically, we expect that samples from traditional population is high in Prevotella and low in Bacteroides, and the opposite is true, for example, in the industrial populations. So the takeaway message here is that the samples from Denpasar are unique to the distribution pattern of these two genera, and it may reflect the status of their community that's undergoing transition in lifestyle. So on this slide, I'm showing a figure from a preliminary analysis of the metagenomic data that we generated about early last year. And I have highlighted the samples from Bali here in the blue box. And I would like to note that in this particular analysis, we only have a data from 11 individuals from Denpasar, whereas in the 16S data, we had 41 individuals. In addition to that, to the Denpasar data, we also have data from Padawa, Daikot, and the other communities from Kalimantan. At a glance here, the Prevotella abundance doesn't appear to be different between these populations, but the Bacteroides tell a different story. The Bacteroides here in Denpasar shows a greater variance in comparison to other populations. And also, one interesting thing to note is the populations in the Punan population from Kalimantan have a very low abundance of Bacteroides, but they're from just these two genera, it doesn't look like they have differences in the abundance patterns between these Punan populations. So when I analyzed not the relative abundance data, but the presence and absence of the data of OTUs in the data, we found that the Basab community from Teluk Sumbang, shown here on the left file and plot here, are clearly very different from the rest of the populations in Kalimantan, which are the Punan. And there are also significant difference between the community in Rian Tubu and the community in Respen Tubu, which is interesting because both of these communities are sharing close ancestry with each other. On the other hand, there are no significant difference between the Punan in Respen Tubu and Punan Aput in Long Sule, despite the fact they have quite distinct lifestyle. So, and Respen Tubu is the only peri-urban community with low hunting frequency, while Sule is the one with the higher hunting frequency, and it's more remote. So when I analyzed all of the OTUs, not just Prevotella and Bacteroides, I found that there are tremendous difference in the microbiome between the Punan group and the Basab Balinese group. So the Basab Balinese group is on the right side of the ordination plot here, while the rest of the populations is on the left side. And the principal coordinate analysis here showed a striking 43.7% difference along the first axis of the principal coordinates, and about 10.7% difference along the second axis. On the lower right, as a comparison, I put a result from a similar analysis that used relative abundance data instead of present and absence. And we can see that the separations between the Punan and the other appears to be more discrete in this one, but actually the first principal axis only accounts for like 21.7%. Sorry, yeah. Yeah, so anyway, I did some more testing, and it is possible to see this replicate this distinction with just a few strain level of OTUs. And I would not, so in this slide, this is my slide before the summary, and I hope I'm still have fun, but Basically, this is a very preliminary analysis of genetic variation at strain level in the data set. And what I saw here, there appears to be no distinct groupings of samples based on SNPs in these strains. So I'm not sure yet what I'm not, this is a strange outcome, and I'm not yet convinced if this is true, because I haven't really looked at all of the strains that might be relevant. And I'll be happy to discuss this if any of you have an idea on how to approach this question. So in summary, the gut microbiome in Indonesia is diverse, and our analysis of the Balinese cohort revealed intrinsic and extrinsic differences in the microbiome. In contrast, the Punan cohort appears to be more similar to each other in terms of abundance profile, but there are large differences found in the presence and absence of strains. I would like to do further analysis of the Punan communities to gain insights into the environmental and anthropological factors driving these differences. Lastly, it is important to recognize the structural difference in the microbiome in Indonesia, and I believe that understanding local patterns in the microbiome structure could lead to deeper understanding of host microbe relationships and their associations with clinical phenotypes. I want to end this talk by expressing all my gratitude to my college and collaborators, and research just would not have been possible and half exciting without all the collaborations that we have here. And I want to especially thank Dr. Safarina Malik and Professor Hewati Sudoyot, who gave me the opportunity to be part of their amazing team that study diversity, human diversity in Indonesia. And the work that has been done by this team for the past 30 plus years have been a great influence for my values and my approach to conducting research. Thank you, everyone. I have my email here for anyone who want to discuss on chat or ask or possibly have future collaborations.
Thank you, Clarissa.